All right, so we're going to talk a little bit about regulation and safety. So, regulation in the biotech industry. So, we talked about the drug pathway earlier, and we talked about some different processes. So, there's a lot of things that we can do in biotechnology. And remember, we said that biotechnology is about changing DNA. Remember, DNA makes proteins, and proteins makes everything work or not work. So, since we're doing something that has the potential for great benefit and great harm, right? We need a lot of regulation. Okay. So government agencies, um, FDA, that's the big one, right? So any new product through food or plant falls under the FDA. So anything that I'm doing with food or plants, or we can add in there biopharmaceuticals. Right, we can add biopharma in there. So that falls under the FDA. We have the USDA, right? Right. Plant health and safety service inspections. So even when if I want to get a GMO plant, so say I want Roundup resistant tomatoes. So I've got to get the whole process approved through the FDA. Once I have the product approved, the USDA will come out and inspect my crops. Right? So they'll have to make sure that they're not encroaching the fields, that they're single planting, so that the tomatoes that it produces doesn't produce a viable seed, right? And then I have the EPA, right? So the EPA works with the FDA and the USDA to look over biotechnology crops. So I have the GMO product, I have to go to the FDA, right? That's for my process and product. Then I have to go to the USDA, right, for crop safety. And then I got to go to the EPA for environmental impact. Right? So I got to make sure that these aren't harming the environment. Right? And then don't worry about the ones in India because we're not in India. But those are the kind of the three big ones. If I'm just making biopharma, I'm really worried about the FDA. Right? Maybe a little EPA to make sure that I'm disposing my waste properly. Right? But that's about it. Right, so there's some things that we do, and some of these things will be familiar with some of you and not to others. Right? So we have practice guidelines. So GLPs are good laboratory practices. Alright, so my GLPs are my good laboratory practice. So a set of internationally recognized guidelines for generating data. So good laboratory practice, these are instructions. So what it says is it's instructions for safe and consistent use of materials and equipment. So I'm going to handle all my materials one way, and I'm going to use all my equipment one way. And that's the only way I can use them in this process. And it's a way that it is used in industry. So if I have a mechanical pipette, I'm going to use the same process for using it, and I'm not going to deviate from that. Right? If I'm changing its tip every time that I get something new or I change something, I can't today say, well, we're short on tips, I'm just going to do it a little differently. So good library practice says I use it the same way every time. And that way I get the same consistent results. SOPs, right, or standing operating procedures. So an SOP document, right, so that's how you record things. Right? It says who has responsibilities. Whose job in our lab group is it to go get materials and bring them back to the lab station? Right? So whose job is it to document? Right? Whose job is it to pipette? Whose job is it? So it details everybody's responsibilities. How materials are received in labels. So every time I open something up, it should be labeled a certain way. So the SOP I should have a date on it when I received it, and the date when it was opened, right? And then how it's stored. Is it stored in a refrigerator or a freezer, a dark room? So it tells me how it's stored. A detailed plan of how the study is enacted and reported, And then how data is generated. So this is like your lab notebook. So your lab notebook would fall under SOP, right? So it's very important. If it's not recorded, and it's not recorded correctly, then your data doesn't count. Right? So you could go to submit it to the FDA, and they would say, "Where's your SOP?" Then you show them your SOP document, and they look at how you record your data, and they say, "Well, it's not consistent with your SOP, so your data's no good." And then GMP, right? GMP, right? And what we say in here. Lost my pen again. GMP is similar to GLP, but it's for manufacturing. So that's all you need to know. So it's similar to GLP, but involves a manufacturer. So once I get it approved by the FDA and I start making it, GLP goes to GMP. So when I make it, the process I use for making has to be the same every time I make a new batch. Right? Right. So lab safety. We'll talk a little bit about that. So some abbreviations. PPE is personal protective equipment, which you probably know from the news because we talk about it all the time now. Right. So safety goggles, right, should be worn always. Lab coat, chemical resistant gloves, long pants, closed-toe shoes. Right. So those are all examples of PPE. Right. So you can't be like Mr. Kelly and wear sandals to a lab because that doesn't follow good PPE. Right. So when we get back to the lab, right, there are certain things that we have. Goggles are always. Now the MSDS, I found something I forgot to correct. Right, so the MSDS, Material Safety Data System, or data sheet. Alright, that's what I need to fix. So, the MSDS, the Material Safety Data System. So this is a document, right? So this is our old type of document, listing all the potential and real safety risks and hazards. MSDS, we used to have a big giant book. It was full of everything we used. Now it's all done online. But there's a couple of things you need to know. Right? So you'll see these triangles, and if you were in my classroom, I'd show you I have one on my back door. Right? And it lists the triangles different colors. So the things you need to know is the blue is health. Right? So the blue is health. Right? So it lists any particular health concerns I should have with the material listed. Red is flammability. How likely is it to combust, right, when presented with flame or heat? Yellow is reactivity, right? How it reacts with other chemicals, right? Is it an oxidizer? Is it a reducer? And then white is any special concerns. Right? So white is special concerns. This one has a line through it, so there's no special concerns. Right, so this one has a 3 here. So the 3 would say extremely dangerous, right? Wear full PPE. The 4 here would say it's extremely flammable, right? The 3 says it's a strong shock or heat may detonate, right? There's no special health concerns after that. So make sure you know the MSDS and what the colors in the triangle mean, because you'll be quizzed on them. So BSL, right? So almost all high school labs will use BSL-1. So if we were in school right now, we'd be using bacteria, right? And all the bacteria that we have is genetically engineered to be BSL-1. So most high school labs, most colleges. So BSL-1, right? We come over here, agents that are not associated with disease. So this would be these, right? RG means risk group, right? So agents that are not associated with disease in healthy adult humans. So we usually use the E. coli K12. Right? That's what we use most often. BSL-2, moderate threat. So it could make you sick, but you shouldn't die. Right? Associated with human disease, rarely serious, for which preventive therapeutic interventions are often available. Right? So in college, you might use some of these. BSL-3s and 4s. Right? Those are these right here. Agents are associated with severe disease that could possibly kill you. So usually 3s and 4s, you need to get to a government lab to start working on those. Right? Because they limit the availability to those because of potential harm to yourself and the community. Right, we have general signs. Right? So you need to know what the general signs mean. Right? So we have our fire. Right? And we have our corrosive. We have our toxic. This is danger to the environment. You see the dead fish in a dead tree. This one is a biohazard. And this one right here is radiation. Explosive. Electrical. Oxidizing. Harmful. Right. So those are the basic ones. Then we have another explosive, toxic, another oxidizer, another fire or flammable. I guess I should have wrote flammable up there. Right. Some of them have. There's my electrical right there. So, general danger. Right. So, you have different ones that you need to be aware of. But don't worry about the triangles and the colors. They've changed that with the new system, and because it was too confusing whether it was talking about a reagent or a process. But make sure you know what the pictures mean, because it become important. Because every lab should have those associated with them. Right. Volumetric. So volumetric. Trying to get my pen back, sorry. Alright, so volumetric. It's this used to measure. Graduated. Measuring marks. So that's the way you look at it. So this is a graduated cylinder because it has measuring marks on it, right? This is a flask. It's a graduated flask if it has, a lot of them have a measuring mark at the neck, right? So if it has a measuring mark at the neck, it's technically a graduated flask. If it doesn't, it's just called a flask, right? An Erlenmeyer flask is this one right here, right? It's an Erlenmeyer flask. You can see that it has markings on it, so it would really be a graduated Erlenmeyer flask. Reagent bottle. If it has a lip on, cap on it, it's a reagent bottle. So reagent is the same thing as reactant, but it's used in biology. So biology reactants are called reagents. And it has graduations on it, so it's a graduated reagent bottle. If it just had a bottle with a cap, it would just be a reagent bottle. So those are kind of our big ones to use. Disposing of biohazard material, right? So we have an autoclave. So this is an autoclave, right? I have one in my back room. So it uses steam, right, to kill things above boiling, right? So about 121 degrees Celsius we kill things at. Or we have 10% bleach, right, for our countertops. So every time before you start a biotech lab, you clean your countertops with bleach. And every time before you leave, you spray it with bleach again. Right? And that'll kill anything that's on there. Then the last thing is for today is pipettes. So there are different types. So volumetric, right, has graduations. So this is my graduated, right? So a seriologic is different than a standard volumetric. So this would be a volumetric right here. A seriologic, right, has graduations to the tip. So you can see the graduated and serologic. The difference between them is where the graduations stop. Pasteur, right, is made of glass and does not have graduations. It's just used for transfer, right? So none of these is a Pasteur because you can see that these have capacity marks, right? So that's still a single graduation. This is still a volumetric because it has a graduation mark at the 10 milliliter. Right? And disposable, right? Or DPTP, they'll say in some of your lab. Disposable, the P stands for plastic, transfer pipette. And those are these. You'll notice that this one right here, if you can see in the picture, has graduations. So that's a disposable, graduated plastic transfer pipette. Okay? All right. That was pretty simple, and that's it. See you soon.